Well, good morning, everyone, and welcome. Before I introduce today's speaker, I just want to briefly remind you that today's convocation is just one of a series of events that span our academic year. Our convocation series invites us as a community to engage in broad conversations about our world and our place and purpose in that world. For today's convocation, I just want a heartfelt thanks. This speaker series wouldn't be possible without the financial support of the Sverdrup family and NASA's Minnesota Space Grant Fund. This year's series has been filled with a variety of art and calls for action. It included Krista Tippett's ponderings on the interconnectedness of science and religion, then on to Professor Baylock's brain science on why we choke under pressure, the beautiful and stunning music and dance that was shared with us by the Rose Ensemble and the artists at the Martin Luther King Jr. Day celebration. Most recently, we were called by Sister Peggy O'Neill to get a life. Actually, I think it was an order, not really a call. Many of these videos are online at the convocation website. I encourage you to go visit there. Today, we are joined by Professor Bonnie Bassler, Squibb Professor in Molecular Biology at Princeton University. Professor Bassler has earned numerous awards and accolades, including a MacArthur Fellowship, the Wiley Prize, the National Academy of Sciences Lounsbury Award. She was also nominated by President Obama to serve on the National Science Board, the board that oversees and guides the National Science Foundation. But most importantly for today, maybe, she's been a brilliant and energetic communicator of her science to the public. Her TED Talk online has over 1.5 million views. She has been featured on just about every science TV and radio show. And from all of those kind of notes, my favorite is that her research on communication between bacteria has earned her the title of the Bacteria Whisperer. I'm sure you love that one. Today, she will share with us her research on how this knowledge of the language of bacteria may be used to control pathogenicity of bacteria. Please join me in welcoming Professor Bonnie Bassler.
Thank you. It's an absolute delight to be back this morning. Sounds like a lovely seminar series, and I wish I had gotten to go to the rest of them. It's a big honor to be included among that list of people. And so I'm happy to give you a little insight about what the students in my lab are doing back in New Jersey. And so I'm going to try to make this different from last night, but I still work on what I worked on last night. So I hope the people who were here last night will forgive me. I have two slides that overlap with what we talked about last night to make sure all the students who didn't come last night can actually get up to speed and then learn about our newest research in how to think about bacteria talking to each other, working as groups so that they can actually accomplish tasks the same way we can do bigger and better things when we work as groups, and then trying to interfere with that to make new medicines and products for industry. Okay, so to get started, so here's the two slides from last night. So I assume all the students in the room know what bacteria are. These are the tiniest living organisms on Earth. They inhabit every single niche. You have 10 to the 14th of them in your gut alone, and then you're covered, I know it's really nice, you're covered with them. The thing you know about is on your teeth in the morning, that's a bacterial biofilm. There are 600 species of bacteria in that gunk in the morning. You brush it off, and then the next morning it's back. Right? So that's the one that you know about, but what you need to know is that you are covered with bacteria, and they are good for you. So if you look at the bottom, hopefully you can see these, the bottom part of this slide, what we now know is that bacteria, these primitive organisms, inhabit all of the niches on Earth. And then in the case of you and your body, right, as I just told you, they cover you, and most of the time what they do for you are things that are good. And so, for example, in your gut, you need these bacteria to digest. When you eat salad, when you eat plant food, you don't have the enzymes to do that, so the bacteria do that for you. They also educate your immune system so that you can, most of the time, keep bad microbes out by keeping these good microbes in. They also provide you a lot of your vitamins. That's what allows your enzymes to work to do all these jobs in your body. So the view of bacteria that we're coming to in the 21st century is one where they are these partners with us that are essential for keeping us alive. But of course, there's also bacteria out there in the environment that are really bad for us. So there's all these bacteria that just are usually minding their own business out in the environment, but if they come in contact with a human or with an animal or with a plant, they have all these bells and whistles, these features that they use in the environment. And when those features get unleashed, for example, in the human body, they can make you sick or they can kill you. And so bacteria have these bad personalities, they have these good personalities, but what is central to what I want to talk about today is my lab's question, which is, whether bacteria are doing bad things or they're doing good things, how can it be that they can do anything? I mean, these organisms are eensy-weensy, they're tiny. So how can it be that they can either keep us alive or they can kill us? And so what I want to try to tell you is that the way that they get so much power is by counting each other with chemical words, recognizing when they have the right number of bacteria, that if they all change their behavior in unison, they can carry out these kinds of tasks and be successful, right? So they can't act as individuals, they would never accomplish anything, but if they act as a group, then these tasks become useful. And so the way that that works is through a process that we call quorum sensing. And so what happens is when bacteria find themselves alone, so when they're dilute, the world is big and they are small. And so they want to have the program of behaviors going that's good for acting as an individual. So they're doing certain tasks that are useful to keep them alive. But among the things they do, I hope you can see this, is that they make and release, I hope you can see these little triangles, they make and release small molecules that you can think of like hormones. And we call these autoinducers. And so what happens is when the bacteria are alone, they release these molecules, the molecules float away, the bacteria can't perceive them, and so they act as individuals, and they carry out some set of tasks. But then as the bacteria grow and divide in a particular environment, since all of the bacteria are making and releasing their share of this hormone, the autoinducer, the concentration of this molecule outside of the cells increases in direct proportion to cell number. And so when that molecule hits a certain level, the bacteria perceive that the molecule is there, and what they infer by perceiving that that molecule is present is that they must have neighbors around. And so when they detect the molecule, all of the bacteria in unison change their behaviors and they begin to act as groups. And so the reason that I drew these blue is because we talked about this last night. This was originally discovered in bioluminescent marine bacteria. And so what they do together is turn on light, and the light is blue. And so what was fantastic about that bacterium is that it allowed the field to discover this process called quorum sensing. So bacteria are invisible. Right? You can't see them. So if they're acting alone, acting together, how would you know? You can't see what they're doing. We simply knew that they could be pathogens. And so what happened is when we saw that these bacteria make this beautiful blue light only at high cell number, it gave us a visible way to see this invisible world. And so we realized that bacteria actually had the capacity to act as groups. And before that, it was always thought that they were too primitive, they were too stupid, if you will, to be able to do that. And so this blue light allowed us to realize that bacteria could act as groups. And then what happened is once that discovery got made is that lots of scientists started to look at their pet bacteria and ask, could it act as a group? And what we found in the last couple of decades is that probably all bacteria have these chemical communication circuits, and the kinds of behaviors that they control with this process are ones that you need lots of cells working together, and then the process becomes successful. So what we now know is that bacteria can't be pathogens if they don't have this communication process. They need to act together and collectively release all of these poisons that make a person sick. Otherwise, if they act alone, they don't have any influence. So now we get that this process is central to the kinds of behaviors that we actually care about that bacteria do, both pathogenic ones, and then what I'll get to at the end of my talk, we also use bacteria to do all kinds of good things. We use them to treat wastewater, we use them to make medicines, we use them to make all kinds of products that we want for industry. And so knowing that in fact they act as groups is useful to help us make high-level production of things that we want in the 21st century. Okay, and so just one more time, this process now is called quorum sensing. right? They have to have the quorum, so they sort of vote with these chemical votes. They recognize when they have a quorum, the right number of bacteria, and then they all go along together and make this group decision to do something. Okay, that's what you heard last night. Okay, so now I want to change and talk about brand new work that's from my lab on a very notorious pathogen called Pseudomonas aeruginosa. And so this is the bacterium put on this Petri plate. You can see the little colonies, and hopefully you can see that this bacterium is green in color. So Pseudomonas has two personalities. I'll tell you the good personality first. This is a bacterium that lives out in the dirt, and it is used extensively in industry because it makes all kinds of secondary products, chemicals, that we need in industry. It's also used to treat wastewater, right? If you've ever seen those ponds, right, that this bacterium will clean up wastewater and make it drinkable again. So it has this good form of itself where it's highly, highly used in industry and in the chemical universe. But it has this bad side, which is if you are immune compromised or you have the disease cystic fibrosis, this bacterium can infect you. So most of us are healthy. We encounter this bacteria all the time. It doesn't hurt you. But if you have cystic fibrosis, it can make an infection in one's lungs, and people who have CF actually die of a Pseudomonas aeruginosa infection because of this quorum sensing process. It sits in this film on the lungs, it secretes toxins, and it kills people with CF. It's also the primary bacterium that hospitals are worried about. So I don't know if you guys are reading the newspaper. You should be. You're all over 18. So there'll be a number of not so subtle messages. Anyway, so this bacterium, if you've heard, right, people, there's this huge problems in hospitals in the United States now, where you go in, to get your kidney stone or to get, your gallbladder removed or whatever your operation is. And then what happens is you get an infection, and it's actually the infection that is much more dangerous than the process you went into the hospital, right? So it's becoming like the 19th century again, where there's these rampant multi-drug resistant infections. Lots of this is because of this bacterium, Pseudomonas aeruginosa. It's really sticky. It loves to make films on surfaces, so it's sort of all over these hospitals. And so what happens is you go in for a certain procedure, and if you need to get a catheter, you need to get a breathing tube, you need to get some kind of under your skin implanted medical device, what happens is you get a biofilm of Pseudomonas aeruginosa, and that's the problem that actually people get sick from with these secondary infections from hospitals. And that's because it's all over these surfaces, and so it gets transferred from room to room to room. And it affects immune-compromised people. Those are people in the hospital. Okay, so the biofilm thing, the sitting on surface, that's really good if you want to make chemicals and if you want to clean up wastewater. It's really bad if you're in a hospital. Okay, so I don't want you to just think this guy is bad. It's good and bad. And so we want to know if we can do something about those issues. Okay, so first let's talk about the bad side of Pseudomonas. Okay, so one more time. So the way this works for Pseudomonas now is that low cell density Pseudomonas is not making a biofilm, it's not virulence, it's just going about its business, for example, out in the dirt. It's making and releasing these hormone molecules, these autoinducers, and when it's dilute, so when there's not very many bacteria together, it is not a pathogen. It doesn't have the genes on that make the poisons that make you sick, and it doesn't stick to a surface. But when the bacteria grow and divide and they make this autoinducer molecule, when they perceive that molecule is there, that tells them they have lots of neighbors around. And so as a unit, they sit on surfaces, they express adhesins, they sit on surfaces, they make the film, and then all together they secrete virulence factors. So only as the group. And so now I want to talk a little bit more about exactly the molecular steps that happen in that process. So what happens is Pseudomonas, when it's dilute, so when it's by itself, it's a bacterium that has a flagellum. It's sort of like a tail that moves like a boat propeller. And so it's swimming around in the environment, right? But they're far away from each other since everybody's just swimming around on their own. But then when it encounters a surface, remember, it's this bacterium that loves to stick. So if it swims down and encounters a surface, so this could be a particle of dirt, it could be the sink in the hospital, it could be your catheter. What happens is that then it expresses adhesins, so sort of glue, that sticks it to the surface. And then it starts dividing on the surface. And so now what you can see is since they're stuck to a surface, they're now in not 3 dimensions, they're in 2 dimensions, right? They start making these adhesins, and now they're close to each other. And so then they start to make that autoinducer molecule, and so there'll be a little cloud of autoinducer around them, right? So that's quorum sensing. And that they're growing and growing, the autoinducer is accumulating, and when they're in this sort of microcolony stage, it clicks into them that they have neighbors. And so the next thing they do is that they make what's called exopolysaccharides, so I call it EPS. This is like snot. So it is a polysaccharide, a sugar, a long chain of sugars, and what they do is that they cover themselves in this goo that binds them all together, but it also protects them from the environment. So they sort of make this suit of armor made out of this sugar, right? And then they begin to make what we would call the biofilm, right? So that's why this stuff on your teeth, that's not pseudomonas, but it feels gunky. That's because these bacteria have made this exopolysaccharide, and then they start to grow in it. And they grow and grow and grow, and now quorum sensing is really happening. All the bacteria are making this exopolysaccharide, and so the biofilm grows into this sort of community. And what is important about that is making this exopolysaccharide substance that sort of houses the bacteria makes them absolutely impervious to antibiotics. So antibiotics can't get into that and actually kill the bacteria. It's like a shield. And so this is the problem when you have an infection, is that they're in this film, antibiotics don't work, and then when it grows to this mature state, in unison, what the bacteria can do is they can get their toxins out. So all at once, they start secreting 30 or 40 different proteins that chew up the person's tissue, typically your lung tissue or wherever you have an implanted device, and it's the damage from those toxins that kills people who have a pseudomonas infection. Okay, so the steps are quorum sensing, make the beginning of the biofilm, grow in the biofilm, and then release toxins. And so what we think is if we could actually interfere with the bacteria communicating with each other, they wouldn't understand that they're next door to each other, and so they wouldn't make the exopolysaccharide biofilm, and then they wouldn't secrete these toxins, right? So couldn't that be a new kind of strategy for making medicines, right? Make bacteria that can't talk or can't hear, right? And then they don't understand that they're in a group, and maybe we could think about a new way to help people in the hospital. So we wanted to try that, and so we do lots of chemistry in my lab. We've actually discovered, the students in the lab have discovered many of the molecules that these bacteria talk with, and so on my slides, I've been using this triangle to represent this autoinducer, the hormone molecule, but this is the real molecule. It's a ring, and then it has a few, these are just carbons on it. And so what we did was using synthetic chemistry, we just thought, let's change the structure of this molecule, right? That molecule turns on quorum sensing, and so what we did using chemistry was we made a molecule that looks kind of like the real molecule, but it's what's called an antagonist. It shuts off quorum sensing. So the molecule we made, if you look at this one, right, it's a ring. The only difference is this is an oxygen, this is sulfur. So this shape in 3D is pretty much the same. But then our triangle, by my cartoons, has a big bump on the end, right? So it's the molecule plus a bump. And so if you think about how molecules interact with receptors, right, they fit in like a lock and key, right? So this molecule fits into the partner receptor, sends quorum sensing information in. This molecule fits into the receptor, but it doesn't send the information in, and it actually blocks the receptor from recognizing the real molecule. So this molecule is an inhibitor. It's an antagonist of quorum sensing. And so now I want to prove to you that it is an antagonist, and I want to prove to you, or at least start to think about how one goes from understanding that bacteria talk to each other, from beginning to make the first inhibitors, to how one would go to making something for industry or something for medicine. Oh no. There's actually a picture there, a very beautiful picture that you guys are supposed to gasp and say, can't see it at all. Oh, crap. Okay, so anyway, so what happens is that, I'll just tell you this, is that, remember I told you the first step in this biofilm formation and in infections is for the bacteria to sit on the surface. And I don't know if you can see at all, there's sort of it looks like a sort of a star in here. I don't know if you can see that at all. So what happens is Pseudomonas sits on a Petri plate. It thinks it is a surface. And then it moves out over the surface in a process called swarming. So in 2-dimension, it moves out, right? And that is the beginning of biofilm formation. And so this would be Pseudomonas making a swarm, and then, I don't know if you can see here, if we add our inhibitor molecule, the bacteria just sit in the center of the plate and they can't move. So the first thing that I was supposed to show you is that, in fact, this molecule stops the bacteria from swarming. So that's important for making the biofilm. Right, so if you remember what I told you before is they make the biofilm and then they secrete their toxins. And so remember I told you that pseudomonas is green, and that's because it makes a toxin. It's called piocyanin, and this toxin is what kills human lung cells. It actually poisons different enzymes that use oxygen, and that's what kills human cells. And so when Pseudomonas is at high cell density, it secretes its toxin, and so the bacterium is dark green. If we make a mutant, right, so we just knock out the quorum sensing genes, what you can see in this test tube, right, is that the bacteria are not green, they're white. So that shows you without quorum sensing, they can't secrete this protein, which actually has this green color to it. So if we take wild type Pseudomonas and then we add that molecule, that inhibitor I showed you, what you can see is that the bacteria are colorless. So that shows you, on the first slide that you couldn't see, we can stop swarming by adding a drop of our inhibitor, and we can stop them from secreting their toxin. So this is starting to look pretty good in the sense of thinking about, at least in a laboratory setup, how to interfere with quorum sensing. So then we wanted to try to up the game a little bit. And this is the work of a young woman who just got her PhD. Her name is Colleen O'Loughlin. So we would like to know not if you can stop quorum sensing in a test tube, but we'd like to know if you could stop that in a human. However, you have to actually do a lot of steps to go from test tube to a human. So to sort of march our way up that chain, what Colleen decided to use was to use an animal called a nematode. And so these are worms, and maybe in your biology class you might have studied these. These are used all the time for studying neurobiology and sensory perception, right? So these are a model organism. They're called C. elegans. And what's interesting about worms, right, is that they're fantastic for studying how development happens. But from our point of view, what's really interesting about worms is that they eat bacteria for a living. So nematodes live in the dirt, and all they ever eat are bacteria. And so what Fred Ausubel at Harvard showed is that if you give the worms, you know, some innocuous bacterium, it'll eat it and get calories. But if you give the worm a pathogen, it has to eat it, but then it gets an infection and it dies. And so what's so fantastic about these is that they're tiny. You can do lots of experiments very cheaply before you start actually working in higher mammals, right, to test whether your pathogen is a pathogen and whether your therapy is actually a therapy. So Colleen thought, well, let's try to use this worm and see in a living organism, not a test tube, whether or not our anti-quorum sensing strategy can work. And so now what you're going to look at is this is how many of the worms are alive over time. And what you need to know is that a worm will live 2 weeks. So if we just put it on an innocuous bacterium, that's lunch, what you can see is that all the animals live, right? So they would live out for 2 weeks. But then if we take away the bacterium that's not a pathogen and we put the worms on Pseudomonas, what you can see is that all of the worms die within 20 hours. Okay, so Pseudomonas is a pathogen of the worm. So now if we do this experiment, we give Pseudomonas, but we also give that inhibitor molecule, what you can see is that we turn Pseudomonas from a pathogen into lunch. Right? And so now what you can see is that we can cure these worms from this infection simply by adding the molecule along with Pseudomonas. So that looked very promising in terms of the kind of things that Colleen is trying to do. And so that's wonderful if you care about saving worms, but in fact, we actually would care more about saving humans, right? So then again, she wanted to step it up. And so the next thing she did was she said, well, what about cells that come from mammals? So remember I told you that Pseudomonas kills people who have cystic fibrosis. That's this disease of the lung. One can have lung cells, human lung cells, in tissue culture. So you can grow these in the laboratory. And so Colleen grew, these are human lung cells in a Petri plate. And what she did was she developed an assay that when the lung cells die, if they pop, they take up a fluorescent dye, that's shown on this picture, right? And then you can just measure the fluorescence. So if the cells die, if the cells DIE, this fluorescent DYE, gets into the cells, and you can just measure fluorescence, right? So what she wanted to know is, these are healthy cells, a few of them die. If she gives pseudomonas, what happens? Do more of them die? It should. Pseudomonas is a pathogen of the lung. And then what happens when you give our molecule? And so here's Colleen's result. So now what you're looking at is the human lung cells given wild-type pseudomonas, right? And we're measuring fluorescence. So you get a certain amount of death. If you don't put pseudomonas in there, right, all of the lung cells live, right? So less fluorescence. But now, if you give them wild-type pseudomonas plus the inhibitor, it looks as if you haven't put cells there, right? So now what I hope you've seen is in a test tube, we can shut down quorum sensing. In a worm, we can shut down quorum sensing. And now we can keep pseudomonas from killing human cells. And so the experiment that Colleen has started to do is to move this up, and I talked about this a little bit last night, into mice. So there's a mouse that has cystic fibrosis, so people have developed a mouse model that mimics the disease CF. And so what we've been doing is if you give that mouse pseudomonas, it will die. And then if you give the mouse pseudomonas plus these inhibitor molecules, we've kind of got the mouse, as I told you last night, to live. But we have to give, kind of, right? But we have to give a lot of this molecule, right? And it doesn't stay exactly where we want it to stay, right? And it doesn't last the right time in the mouse. And so what we're doing now, and this is where I ended last night, is we're trying to take that molecule that I showed you and refine it. We have to make it much more potent so that you can give a smaller quantity. We have to make it safe. It's not, you can't let it just go all over the body and do harmful things. And then we also have to make it look much more like a drug so that it could be delivered. And so that's the state of affairs in terms of thinking about this process. But at the time that Colleen was doing these experiments and thinking about this molecule, thinking about Pseudomonas, a physicist named Knut Drescher joined my lab. Okay? And so he had never worked on a living organism before. He was a fluid flow dynamics. I know this is organized by the physics department, so I'm going to try and fit in. Anyway, so he was a fluid flow dynamics. So Knut studied how does flow happen. You know, how do you get turbulent flow? How do you get laminar flow? All kinds of crazy stuff. And he came to the lab and said, you know, so he's a postdoc. I've studied physics, but I really want to study biology. So he had never held a pipette in his hand. He'd never worked with a living organism or a gene or anything like that. And he said, I want to think about how physics plays out in biology. And so he knew that we were working on this project that I just told you about. And he said, you know, Bonnie, it's really fine, you know, if you add these molecules in a tissue culture, you add them in a flask, you add them in a test tube. Under those conditions, all the bacteria, if you shake them around in a flask, all the bacteria perceive the exactly identical environment, right? And so then, of course, you get these nice results with the experiments of Colleen's. And he said, what I think that you should do is to move these experiments from a flask, like if you're really going to deploy these molecules, if you're really going to make something that's going to work in the world and not in a lab, you have to stop working in a flask and actually start working in situations that more realistically mimic the natural world of a bacterium. And that situation is a biofilm, is not shaking around in a flask, is not added to tissue culture cells. So remember, I told you about the steps of this biofilm formation, and these bacteria, you know, grow into these 3D films, right? So that's part one. They're in 3D. They're not just shaking around in a flask. So the guys, you can imagine, the guys at the top perceive a different environment than the guys in the middle than the guys at the bottom, right? So that's part one, is it's 3 dimensions. And then the second thing he said that's wrong with your, he's kind of obnoxious, it's wrong with your experiments. I would like to say that I did have a nice career before he joined the lab too. But anyway, the second thing he said is wrong from a physicist's point of view is things don't shake around like that. The world works under pressure-driven flow. And so remember, he likes flow. So what I mean by that is the way your kidneys work, the way your heart works, the way rainwater works, is that flow of all, like air, urine, water, flow happens, right? And there's pressure that drives it, so it's intermittent, right? And there's always flow. Things do not shake around in flasks. And so he said, you need to start thinking about these biofilms not simply on a cover slip, which is how everyone in the field had grown these. We just seed the bacteria on a cover slip and then give them some nutrients and allow the biofilms to develop. He said, you have to think about these the way the world works. And what that means is not flat. It means 3 dimensions, curves, eddies, bumps, grooves, and pressure-driven flow. And so what he did was to build a microfluidic device that if you make macroscopic world small and you want to know what does space look like, it simply means curves. So he built this device, right, that has all these S curves, right? So that's supposed to look like bumps, like what would be in your lungs, what would be like through dirt particles, what would be in a water filtration device. And then he can attach a pump here, and he can flow liquid through those devices, right, under pressure, like the rainwater, like your heart. And so when he did that, and then he would put pseudomonas into this end, what he saw is if he measured the rate of flow that comes out of the right-hand side over time, everything was fine. And then at about 50 hours, with no warning, the flow would stop, right? It went from flowing fine to nothing within 10 minutes. So it was catastrophic, and there was no warning. And so he did this over and over and over again. And of course, I wouldn't show you this if it didn't always work like this. And so then he brought these results, and we started to talk about these. And we said, whenever doctors talk about stents, or people that work in the water sewage treatment plants, or people that work on cooling towers, whenever they talk about what happens in their system, they say everything is fine, and then your stent just fails. Everything is fine, and then your catheter fails with no warning. Everything is fine in the water sewage treatment plant, and then flow stops. And no one has been able to explain why. Because all of the models look like this, where we have the bacteria on a flat surface, and then they simply grow in this biofilm. And if you actually do the math, like if you had a pipe or a catheter or a stent, and you do the math, like how does a film flow from grow from one side to the other, it would take eons if it was just growing in like this. And so Knut had the idea that maybe if it's more about these curves than about these flat surfaces, maybe physics could explain to us why these processes fail. So he took this device and he put it under the microscope. And I'm going to show you a movie in 1 second. And he flowed pseudomonas in that we have labeled green for a little while. Then he's going to change the reservoir, and he's going to flow pseudomonas in that are labeled red. And so I hope you can see this. And I hope what you can see is that green is on the walls. And then what I hope you can see is that red comes into the middle. And so what he showed is that the way it works, if you have corners, right? So this is just a close-up of a certain part of that device. What you can see is that, remember, green cells are flowed in first. So those cells attached to the wall, they're going to make a biofilm. Quorum sensing kicks in, right? And then what happens is they make that exopolysaccharide. And remember, I talked. So it's like a fishing net. It looks a lot like my hair, actually, right? So they make this biofilm and this exopolysaccharide, and then when you actually have corners instead of a flat surface, what happens is that this goopy net gets caught. I hope you can see this with the light, to corner, to corner, to corner. And then it's red. Remember, I told you put green cells in first and then red cells in second. What happens is that the green cells make the exopolysaccharide. It acts like a fishing net, and it just gets caught from edge to edge to edge. And then anybody who's coming downstream, sort of like salmon swimming, right? The cells that come in, they simply get caught in this net. And so what happens then, and we believe that this happens in industry and in medicine, is that the bacteria initially attach to the walls of the device, right? They start to make a biofilm, they make this EPS, and then anything that comes down gets caught, like cholesterol, like blood cells, like dirt, like gunk. And so we showed that this is true because he can put the green cells in, and then we can just add beads, not even a living organism, and we will clog the device. And so what we think is that by changing this model of biofilms from these bacteria growing on this flat surface into one that mimics the physical space in which bacteria really live. I mean, everything has curves in it, everything, right? Right? That maybe what he's found is why these devices fail catastrophically. It takes time for the bacteria, remember the 40 hours, to grow on the wall, for quorum sensing to kick in, to make the exopolysaccharide, and then after that, it's instantaneous. Anything that comes down the pike gets caught, and then at a certain time, all these devices probably fail. And so then remember Colleen? So she gave Knut her molecule, right? And he flowed that through this device, and you never get the clog, right? So now what we're trying to think about is, could Knut, the physicist engineer, embed Colleen, the chemist biologist, molecule into these kinds of devices to make inherently biofilm resistant devices? So that's what they're trying to do. But then one other thing before we did that, I said, we said, Knut, this is great. Now we have a new model. It looks different than everybody putting it on a cover slip for the last 20 years. Maybe that was a goofy model, but is our model any better? Like, do those films, those bridges that go edge to edge to edge, happen in anything other than the device that we made? I mean, it's still in the lab. Okay, we put curves in it, but it's still in the lab, right? So does this really happen? And so I'm going to just show you, to finish up, 2 little examples. So the first thing is, remember I told you that these water filtration devices fail, right? They get all clogged up. I mean, in this case, pseudomonas is good, but then when this biofilm forms, these water filtration plants close down for unexplained reasons because flow stops. So these water filtration devices are just a spiral wound mesh. Maybe you guys have seen these probably on the hallway of the biology department. And so they're just a mesh that's lots and lots of corners. That's all that is, right? So Knut, we used to have one of these on the floor of my hall too, but Knut cut it up and put it under his microscope, okay? And then we labeled the pseudomonas in green, and so this is the mesh, and I hope what you can see is going edge to edge to edge to edge are all of these green pseudomonas bridges. And then sure enough, this thing clogs. And so we think we found this bridging business that happens, at least in this kind of a setup. The second thing is that we got a company to give us a stent. And so you guys are too young, but this is what a stent looks like. This is one that goes in for a urinary stent. It's just a bunch of corners and bends. And so again, he put this is a pity you can't really see this. He put this under his device. Hopefully you can see under the microscope. The stent is there, and then he's got a close-up going edge to edge to edge, corner to corner to corner, are all of these streamers of biofilm. So what we think then is that the biofilms don't start from outside in, they start from inside out, and that is why they clog medical and industrial devices. And so what we are trying to do right now are these 2 things. We're trying to make the molecule, as I told you in Colleen's experiment, much more medicine-like, and then we're trying to think about, maybe you'd like to make a stent that simply has fewer corners in it, right? Or maybe you'd like to actually embed with the engineers, working with the chemists and engineers, embed anti-quorum sensing molecules into stents to keep them infection resistant, but then going back to Pseudomonas making biofilms that are good in nature, where we try to get them to make products, maybe you'd actually like to seed these biofilms instead of on a flat surface, on a curvy surface, so that the biofilm could form better, right? And so we're trying to work this both anti and pro. And so then to finish up, again, we're making the molecules much more medicine-like. We're also screening for other molecules. We're trying to make these biofilm-resistant materials, and then, of course, last night and tonight, I just picked this one pathogen that we work on, Pseudomonas, to tell you some examples, but in fact, my lab works on lots of different pathogens, and now also lots of bacteria that are good in the environment, and we're trying to think about these strategies in both of those cases. And then I want to finish by telling you about my heroes. So Colleen O'Loughlin, I just wanted to tell you that she started in my lab as a high school student. She was a chemist and was interested in chemistry, and she came as a high school student, had no plans to go to college, but then she decided to go to college, so she went to Ithaca University, which has a very small research program, but enough that she could actually go into the lab, and she made in Ithaca, she made some of the first anti-quorum sensing molecules as an undergraduate ever made. And then she called me up one summer and said, could she come and test those? And so she came and tested those, and then I kind of kidnapped her and said, you need to graduate early. So she graduated in January and came to my lab as a technician. Then she enrolled in Princeton and got into our program, and she got her PhD last week. So all of that was 4 years from when she was in college. She's now doing a postdoctoral fellowship at UC San Francisco with a really fantastic guy that works on the microbiome. right? And so I just want to say that this is a person who grew up in a family of people, she has 7 brothers and sisters, none of whom ever went to college, right? And so she went from high school to PhD in 9 years, right? And so if you don't know, she was not a person that knew she wanted to be a scientist, right? But I think, just trying something, right? And you can go on this unexpected adventure. This person, Knut Drescher, he's still in the lab. He just got a job offer at Cornell. So remember I told you he's a physicist. So this is a person who had never pipetted a bacterium or actually held a pipette in his hand. He made all of those strains, like marking those pseudomonas with green and red, and did all of the experiments, just started to think like a physicist about how physics plays out in real biological systems. And he had the courage to come to me, and apply for a job, and just said, I don't know how to do biology, but I know physics. And I said, well, I know how to do biology, and I don't know physics. And that actually might be an interesting interface. And so he came into the lab, the 2 of them teamed up, and then I told you just a few of the stories that both of them had. But I think if you do want to be a scientist, right, if you know that, don't get stuck where you are, right? Science, these disciplines, the traditional disciplines are gone, right? Having physicists try to think about how physics problems work out in biology, or having biology try to think about how physics might play a part is really, really fun, and I think it's the future of how science is going to happen in the 21st century. Likewise, Colleen did hardcore synthetic chemistry, and she also made lots of mutants, right? And so having a foot in these 2 different worlds just makes everybody do things better. And so I think if you're a curious person, even if you don't have the confidence, you don't think that you know how to be a scientist, if these kinds of puzzles and figuring out how the world works appeal to you, you should just try it, right? Somebody taught me to pipette, someone will teach you, right? And so if you go to a lab, right, you have to remember that we all believe that someone invested in each of us when we didn't know what we were doing. And so there's a commitment to invest in the next person. And so I think the other thing I would just love to say, maybe you guys are all scientists, is that we also have this bad stereotype that we just sit in a dark room by ourselves. Not true, right? Labs are big, huge rooms, filled with young people. The music is playing. They're talking about the Final Four, right? They're, doing, and then occasionally they do experiments, right? And so I would just tell you, right, encourage you, right, to actually not believe what you've heard about what it's like to be a scientist, right? So if you don't want to sit at a desk and you don't want to type, and you actually want to think about questions that no one has ever thought about before, and you want to do it in a group of your peers who are smart and curious, you should try science. Thank you for having me here.
I'm curious to know whether your inhibitor has been an inhibitor for all of the different species biofilms or just pseudomonas?
Right. So, okay, so here are the questions. So for, last night I showed that each bacterium uses I said I would repeat it, and then I didn't. The question is whether very short attention span, is whether the molecule inhibits only pseudomonas or inhibits all biofilms, right? And so what I didn't talk about today is that remember the molecule I showed you that pseudomonas talks with? Each bacterium species talks with the molecule, but it has a slightly different tail. So the molecules are exquisitely specific. So Pseudomonas talks with its own molecule, Vibrio talks with its own molecule, listen to me, Agrobacterium talks with its own molecule, right? And so each of those is a little bit different. And so this molecule that we made, so you would guess maybe the inhibitor would be very specific. So it turns out it's not. It's very non-specific. So somehow putting that bump on the right-hand end of our molecule enables it to slot into the different receptors, which all kind of look alike. And as far as we can tell, it shuts them all down. We've tried that in 4 species, not hundreds, 4. So it's super generic. It's not species specific. That's what we know so far.
I was wondering if you've tried introducing an agonist to
You guys
induce early virulence.
Right. Okay, so I don't have to repeat that, but I'll do it anyway. I'll tell you. So that's a nuance, right? That's a great question, too, right? So if you think about how quorum sensing works, right, what I told you, right, these molecules accumulate, and then the bacteria launch their virulence factor program, right? So my strategy is make them so they don't realize the molecules are there, so they never launch. Your strategy, which I think might work even better, is what if I trick the bacteria? Like if I add a super autoinducer, right, an agonist, early on, then the bacteria like think, oh, there's the neighborhood here, and they start secreting their virulence factors when they're at low cell density. That is a huge red flag to the immune system, right? So I think this is optimized. You know, the bacteria count to the right number that the task is going to be successful. And so if you either make them not count or make them show their stuff early on, the immune system gets rid of them. And so we have not found that molecule yet, but we did it with mutants. So we can make a mutant that believes, if you will, it's always in high cell density mode, right? So it just doesn't need the autoinducer. It always has its quorum sensing circuit on. If you put that in a mouse, it's avirulent. Just like if you put the non-quorum sensing mutant in a mouse, it's avirulent. And so I don't know which strategy, if any, is going to work better, right? And so we have a pony in the race of trying to find pro-quorum sensing molecules and anti-quorum sensing molecules, and we're just going to work them and let nature tell us what's the way forward. And it could be in different scenarios, one works better than the other, but I think that's a totally profitable avenue of research. We're on that, right?
So my question is, by the way, great talk, by the way.
It's a silver-tongued devil, yeah.
So does...
You have a very bright future. I don't really care what your question is. Yeah.
So does having a pseudomonas infection lead to high blood pressure? Just because there's going to be like clogs and all this
Yeah,
stuff?
that's an interesting idea. I actually don't know the answer to that. I think that when it's in your lungs, right, you see what he's saying? He's saying it's going to make a biofilm and it's going to clog things up, right? So that's an interesting question. Like if you had and I don't know the answer to that. I think in your lungs, it doesn't, because that's not about blood flow. People that have the CF biofilm in their lungs, they can't breathe, right? And so that's an interesting question. Like if you had these catheters or things like that, does it cause high blood pressure? I don't know, and I don't think anybody's looked at that. Yeah, so I don't know.
Any other questions?
So there has been some data that has shown that the non-CF lung, or in general, won't get the pseudomonas infection. There's ideas that it might have something that's an inhibitory compound that the CF lung doesn't have. Do you think it could be your molecule?
So it's not that. So right. So we don't. This doesn't happen to us. So we have two things. One, we are better able to breathe, to clear our lungs, right? But also, what if people that have CF can't make this molecule, that there's tantalizing evidence, right, that keeps pseudomonas down? It is not that molecule because that's a synthetic molecule, right? And so I think no one knows what the molecule you're talking about is yet and whether or not it impinges on quorum sensing. But we were talking about this last night, the idea that the host either makes molecules that promote good bacteria to do what they do, or that makes molecules that keep harmful bacteria from doing stuff. I think that could be all over the body. And now what people are looking at is, besides bacteria talking with bacteria, what about a dialogue, an inter-kingdom dialogue between beneficial bacteria and the host and also hostile bacteria and the host? And if you could find those molecules that naturally exist and then use those as the starting blocks, right? Everyone is hunting for those kinds of things, right? But in defense of my field, right, it took us, people knew about bacteria for 400 years, and we only discovered 20 years ago that they talk to each other. So it's really, young, right? We only know these few molecules, right? And so now this realization that these could be really complicated chemical dialogues, I think things will happen faster, right? Because we get it now, at least at some level, that this kind of a treasure trove of molecules that are doing all of this behavior modification out in the world, right, they exist. And now we just have to be clever to go hunt for them.
I was just going to throw in when bad breath will be a focus of research.
It is.
So I was just curious, as far as like the medicine-like, making the molecule more medicine-like that you were talking about before, what exactly was it that was causing the mice to live, sort of?
Yeah. So we don't actually let them live, right? And so So the molecule we have, the one I showed you, there's an enzyme in lung cells that cuts that ring off so humans, mice, have an enzyme that's called a lactinase that opens, remember my molecule has 2 rings. It actually opens this ring. That's just an enzyme that we have, right? So the molecule I showed you doesn't last long enough to be a drug, so that head group on the left side must be changed, to something that you don't have an enzyme in your lungs that opens it, because then you have to constantly give them, right, and a lot of it, right? And so the mouse does better than the one that isn't treated, right? So we're really confident about it, except that it's not a drug-like molecule. And so medicinal chemists actually have a lot of chemical tricks. They're sort of standard in a nice way, where they build in features that make molecules impervious to the enzymes that we have, that make molecules, like you don't want to give a molecule that you want to give it to lung and then it goes to your kidneys and gets excreted, right? They make molecules that stay where you want them to stay, but not stay forever. So all of these features, right, that you don't know, you take a medicine, and but these features over 100 years, chemists have learned to build into molecules, starting molecules like the one we have, the features that make them look like a medicine. Right, which is they go where you want them to go, they're extremely potent, they're selective for the bacterium, and they don't harm your cells, and they don't get degraded by your cells. We have not done that here, right? And in defense of my gang, right, why would we do that if it was never going to work, right? So we had to try the molecule we had first, and that gave us the confidence that there's merit to the idea, and now we really should invest a few years in actually screening for other molecules and making them look like medicines, right, based on these initial results that I just showed.
Any last question? How about we thank our speaker
Thank you,
one
you
last
guys.
time?